Okay, so this is a little screencast video to help out with this project on proteins and enzymes and their structure. So right now we're at this website, rcsb.org, which has the protein data bank in it. So you'll want to come to this website and enter your search term. You want to use the name of your protein. I'm going to use dehydrogenase as an example because it will allow me to show a bunch of interesting things. So when I do my search for dehydrogenase, there's not just one dehydrogenase. So this is structures of multiple versions. There's human, there's rat brain, and we're just going to use the first one, the rat brain, because it shows several interesting facets of the structure that are things you're going to want to include in your presentation in your project. So we click on this. There's a lot of technical information here that you're not going to be so interested in for your project, but if you want to learn more about it, you're welcome to play around. You can see where this structure was published in the literature in the year 2000 in the Journal of Molecular Biology. But we're interested in the structure, so let's go to the 3D view structure. And I'm going to scroll down. So a few important features you want to see. First, you've got the overall 3D structure in color here. And if I grab this and rotate it a little bit, you can see there are 4 colors. And that corresponds to this protein having 4 subunits. So the quaternary structure is that there are 4 subunits that all come together. We've got this one here, subunit A. So you can see in the little box that popped up at the bottom as I went over the green subunit, it's got a capital A in there. You can also see that it's highlighting, right? There's that little piece of the spiral highlighted. That's actually a specific amino acid in the chain. It's highlighted in pink on the green subunit. It's highlighted in pink on the very top in that long list of letters. And then down in the little box at the bottom, it's there again, and it's also another A. So what is that? So that A is the subchain. LYS in the box at the bottom is the code for lysine, right? So that's one of the 20 essential amino acids. And then up here in the chain up here, it's got the letter K because K is the 1-letter abbreviation for lysine. LYS is the 3-letter abbreviation. So you got several things going on here. So first off, this part at the top is the primary structure of your amino acid. And it goes primary structure of your protein, amino acid by amino acid, starting at the N terminus and going all the way down to the C terminus. And as you go along here, it tells you in the little box at the bottom, as you go from one to another, it tells you, oh, amino acid 15 is threonine. Then amino acid 16 is glycine. Amino acid 17 is another glycine. And then number 18 is an alanine. So this up here is your primary structure. For your project, I'd like you to give the whole primary structure from here and then explain like one segment of it. And it can be a segment that you think is interesting or that you just pick because it's the beginning or the end, or maybe it's a spot where an important mutation sometimes happens, right? There's mutations in enzymes that cause different diseases, and maybe there's one that's important in your enzyme that you want to mention, and that's the piece of the primary structure you want to tell us about. So that's primary structure up here. Then we talked about quaternary structure. We have the 4 subunits down here, A, B, C, D, and when you highlight your cursor on one, it tells you if you're on part A, B, the pink one is D, and this blue purple one is C. Now something another thing that you should see here is you can see a lot of spirals. Those are alpha helices. Maybe a little less obvious, I will zoom in a bit, is that in addition to spirals, there we go. Let's see, you can kind of see it there. Let's see if I can. So right here, we have this arrow that's kind of flat. That is part of a beta sheet. Right next to it is another arrow, another beta sheet part, and then a third one here, and a fourth one here, and they're all lined up together. So we've got beta sheets going on in the secondary structure of this molecule, of this enzyme, as well as the alpha helices like this one, right? And as I trace it around, on that bottom box, it tells me which amino acids are in that alpha helix chain. Maybe you want to choose one of these to talk about the primary structure and tell us which amino acids are in that little piece of alpha helix loop. Maybe you want to tell us the set of amino acids that are in this one piece of the beta sheet. Something that you might be wondering about, all these little dots floating anywhere. Those are oxygen atoms that are in the water molecules that were part of the crystal that this enzyme made when it was crystallized to figure out its structure. We can actually remove those by clicking on the eyeball next to water. And then we have ligand. So ligand in this case is actually pretty interesting. I'm going to zoom out for a second and rotate a little bit. So the ligand's in here, and here's when I can put my cursor on. See if I can zoom in on that a little bit. So this ligand here, when I put my cursor on it, turns pink. That is nicotinamide adenine dinucleotide, NAD, which in the video on vitamins and minerals, cofactors and coenzymes, this is a coenzyme, and it's one of the B vitamins that you need in your diet. And this dehydrogenase enzyme requires this coenzyme in order to be able to do its job. So not only do we know what it is, we know how that coenzyme fits into the enzyme itself inside its structure. I especially like this structure because it has another little goodie in here. Let's see if I can move my cursor and find it. So that one right there, kind of in the background that turned pink, is estradiol. That's the substrate. That's the reactant molecule that this enzyme catalyzes a reaction of estradiol, right? It dehydrogenates because it's a dehydrogenase, and it dehydrogenates estradiol in its job. So we have a crystal structure here that not only shows the enzyme itself with its cofactor, it also has its substrate in the structure. So we've talked about primary structure, we talked about the secondary structures with the helices and the beta sheets. We talked about the quaternary structure with the 4 subunits. I do want to make sure that you take the time to rotate your molecule and look to see what's the tertiary structure of just one of the subunits, right? These are not all the same. And if you look at it from one side, this looks a little bit wedge-like. If we rotate it, you can see it's sort of globular, sort of wedge-like because the 4 of these all link together. You can see the ends of the protein chains on here, and if you hover over it, it'll tell you what amino acid is at the end of that chain. So there's a whole lot of information here. For your presentations, my recommendation is that you find the interesting pieces in here, you do some screenshots, and you crop things so that you can really highlight the primary, secondary, tertiary, quaternary structure. You might be able to also show any cofactors or coenzymes that are in there. You might even, like this one, get lucky and be able to show the substrate that is part of your enzyme. You can include a link if you want to in your slides to be able to go directly to the protein if there's something that comes up during your presentation. But mostly I think some well-chosen screenshots will show a lot of the information that you want to explain in your presentation and your project. And this is a nice, free, publicly accessible website. Again, if I go back to the home page, it's RCSB. I'm not even sure what RCSB stands for, but it's certainly research something. And it is the Protein Data Bank. I will post a link to this along with the video, and hopefully this is useful to do nice work on your projects.